Welcome back, folks, to microbiology. Today we're going to be talking about chapter 7, which is microbial genetics. So in chapters 5 and 6, we talked about microbial metabolism and nutrition and growth. So essentially, those are the processes that microorganisms are using to carry out their living functions and then grow and reproduce. Now, where do we get the code that allows for us or allows for the microorganism to actually know which proteins it needs for which process, when to express them, how are those proteins actually made, and how are those metabolic processes regulated? Well, that gets at the core of genetics. Essentially, the answer to all of those questions is DNA in, different processes and how it's actually kind of expressed. But the DNA is the genetic information that's held in any cell, no matter what domain it's from, although we'll focus on prokaryotes, and allows for that cell to do the functions that it needs to do from metabolism to growth and reproduction. So I definitely recommend that you review your material, your notes from chapters 5 and 6, and then preview the chapter 7 material so we can go ahead and jump into that content. Welcome back. So what we're going to cover today is we're going to talk about the structure of DNA. Then we're going to talk about how that DNA is expressed by talking about replication, transcription, and translation. And we'll go into those processes in detail. And then we'll talk a little bit more about regulation, mutation, and how bacteria have some really unique characteristics that make them really beneficial in the lab setting. And so a kind of a culmination of those ideas that we'll talk about today is this comic here, where you have one bacterial cell kind of gossiping to another cell right as their friend is there. Don't pick it up, I said. And he says, it's just a plasmid. What harm could it do? Well, just look at him now. God knows what protein he's expressing, right? And so it's interesting how bacterial cells, they have their genome and their genetic information, but they also have unique ways of expressing and interacting with each other at the genetic level that we'll get into in this class. Okay. Now, genetic, genome, chromosomes, plasmids, some of these words that we have already used and will be using throughout the lecture today, let's define them, okay? So genetics is the study of inheritance and inherited traits. So essentially the study of genetic information and how it's expressed, okay? And each at kind of each science has a unit that it kind of works with, right? And so if you're talking about chemistry, you're looking at the unit oftentimes of an atom. And if you're talking about biology, oftentimes you're looking at the unit of a cell, and of course, there are other units as well that we work with, but generally speaking, the unit of genetics is the gene. And so when we're talking about DNA, it's a long molecule that's made up of base pairs, but the functional unit is actually a series of base pairs, and you'll see what a base pair looks like soon, that make up a gene. And that gene is the series or the sequence of information that's actually expressed. And each genome has a number of thousands or hundreds of thousands or even millions of genes that it expresses. And the aggregate of all of those genes, plus the other pieces of DNA that might be non-coding, for example, or not expressed at that moment, are called the genome. So the full aggregate of the genetic information of that individual organism. And so by definition, any cell from any domain should have DNA, should have genetic information in the form of a genome that it is expressing to keep it, to sustain it, essentially. And that is a eukaryotic cell, a prokaryotic cell, bacteria, so on and so forth. And so each cell has that genome as its blueprint or its code. We'll talk a little bit about how those genomes might be different, but they're usually composed of chromosomes, And chromosomes are long DNA molecules that have all of the genetic information within them, and they are single units, they're single molecules, okay? Now, in bacteria and archaea, oftentimes each cell will just have one chromosome, whereas in eukaryotes, cells will often have multiple chromosomes, although there are some exceptions where you actually have bacteria that have multiple chromosomes as well. In prokaryotic cells, these chromosomes are housed without a membrane outside of them, but in a region that's called a nucleoid, whereas in eukaryotic cells, these chromosomes are actually engulfed or encapsulated in a nucleus. Now, chromosomes are the essential genetic components of cells. Like the essential genome is going to be within the chromosomes of the cell, and you can't have a cell without chromosomes. But especially in the case of prokaryotes and in some eukaryotes as well, you also have these additional pieces of genetic information, some of which are called plasmids, and plasmids are the ones that we'll focus on, although there are a couple of other non-essential genetic components within cells. So a genome will include all of that genetic information. If the cell only has chromosomes, then it'll be all of the chromosomes and the genetic information held within them. But it also might be chromosomes plus plasmids, where you have this non-essential genetic information in the cell as well. And we'll talk more about both of these units. Now, another couple of terms that are essential, we won't utilize them so much in this lecture specifically, but they're really important to the discussion of genetics. Genotype is the kind of the set of genes that are held within an organism, and phenotype is the expression of those genes. So oftentimes, and essentially almost all the time, you will not be expressing your full genome, you being a prokaryote or a eukaryote, any organism, will not be expressing their full genome at all the time at any one time. Usually what's happening is the specific genes that are needed at that specific time are being expressed and the other ones are being regulated, okay? And so the genotype is all of the aggregate of the specific genes that are in that organism, and the phenotype is those characteristics that are being expressed. Okay. Now, another set of terms that are kind of essential to this discussion includes macromolecules. So if you remember back to other previous biology classes, there are 4 essential classes of macromolecules, or large biological molecules, that essentially make up cells. And so when we're talking about genetics, we're really essentially looking at the relationship between these 4 molecules, or these 4 types of molecules, where we're looking at nucleotides, generally speaking, are the molecules that hold hereditary information, although there are some exceptions that we have talked about, like ATP. But nucleotides generally are the class of macromolecules that hold genetic information. And those nucleotides, for them to be functional, for them to be expressed, usually need to be translated into proteins. And the proteins then become the functional units that actually carry out the metabolic processes within the cells in relation to other proteins, but also in relation to making and breaking lipids and carbohydrates. Okay, and so when we're talking about genetics, in a very basic way, we're actually looking at the interaction between these 4 types of macromolecules. And so in the second link below, I'll get to the first link in a minute, but in the second link below, there's an optional review video for the amoeba sisters, just to look back over who are the macromolecules, what are the 4 macromolecules, and kind of what do they do. Now, we said that genetics is essentially the relationship between these 4 types or classes of macromolecules. That kind of relationship or association can be boiled down into what's called the central dogma of biology. And the central dogma of biology will, again, essentially inform this lecture today. Now, the central dogma of biology takes what I just said about the relationship between these four macromolecules and gives us a rule that we follow. And that is that DNA, which is oftentimes your double-stranded molecule, is replicated so that organisms can reproduce. But when it needs to be expressed, it is transcribed into a copy. That copy is in the form of RNA, generally speaking. RNA is single-stranded, but it's still a type of nucleic acid. So both of these are nucleic acids. And then in order for that copy to actually be expressed, it needs to be translated into a protein. And that protein becomes the functional unit that goes out and does the work in the cells, interacting with other proteins or lipids and carbohydrates. Sorry, I just knocked my desk there. And so this central dogma tells us that DNA is the basic and essential blueprint, which needs to be replicated so that it can be passed on to other generations, or in the case of bacteria, sometimes other peers. And it can be expressed through transcription and then translation, okay? And so we're going to talk about, in this first part, the structure of DNA, how it is replicated, how it is transcribed, and how it is translated. Okay? So let's start with the structure of DNA. Okay. So the structure of DNA is composed, like we said, in long molecules that essentially hold the genetic information of the cell. Okay. And so when we're talking about bacterial cells, let's get a little bit of perspective. Like, what is this long DNA molecule? How does it look? In an E. coli cell, E. coli is just about 2 micrometers long. So it's a very, very small cell. But inside of it, it carries all of the genetic information that it needs to do its various metabolic processes. Well, that is actually a lot of information. And that is carried in a strand-like molecule, which we said was DNA. And in the case of E. coli, that strand is made up of about 4.5 million base pairs. And I'm going to show you what a base pair looks like in a minute. And that 4.5 million base pairs codes for about 4.5 thousand different functional units or genes. And so all of that information and that E. coli will regulate which genes are turned on or off so that it can do various processes and functions. All of that information is carried within that strand-like molecule in the cell. And we'll look at how those strand-like molecules are organized in different cells. But to give us some perspective, that strand just in an E. coli molecule is about 1,600 micrometers long if we unwind it. 1,600 micrometers long. So the cell itself is only 2 micrometers long, but the genetic information that it holds within it is 1,600 micrometers long, right? To give us, even though we're focusing on prokaryotes, to give us another kind of comparison for humans, we don't just have one chromosome like most prokaryotes do. We have 46 chromosomes, and they're pairs of 23 and 23 from our parents. If we took all of those 46 chromosomes and unwound them and aligned them, that would be about 3 meters long. So 3 meters, about 9 feet, that's taller than most people. And that would just be or all people. That would be the genetic information from just a single cell. And we are multicellular individuals that have tens of trillions of cells, right? And so the genetic component of cells is very significant. And it has within it the blueprint that allows for those cells to do their functions, okay? And so those strands of DNA that we talked about, they're made up of two adjacent strands together that have what are called nucleotides. And those nucleotides are the subunits or components of that genetic information. So let's look at this. Here's our E. coli cell, and it has its genetic information kind of condensed in a specific region without a lot of membrane. You know, there are some cells that have a little bit of kind of membrane folding that allow for this, but not a full-on membrane like a nucleus. And if we were start to kind of unwind and look at what that genetic information looks like, this is what we would see. We would see the DNA, which is what, like we said, very long molecules that are made up of strands or filaments. And there are two adjacent strands in the blueprint, which is DNA. Okay, the two adjacent strands are together, parallel, but then in a helix, in a helical form. You'll see that that parallel helical form becomes really essential whenever we want to replicate that genetic information or make copies of it. Okay. And each of these strands has on it base pairs. Those base pairs essentially are an alphabet of 4 different units or letters, and they are aligned in a sequence or series that says whatever is held in that information, right? And so kind of just like an alphabet, a linguistical alphabet, you can literally read what are the base pairs that I'm looking at and what is that genetic information saying. And so you have A and T, adenine and thymine, that are 2 base pairs that pair with one another, and G and C that pair with one another in DNA. When we make a copy of DNA in the form of RNA, then we switch out the T for a U, which is a uracil, okay? But, okay, let's look at these in a little bit more detail. What are these letters made of, these letters or units made of? If we look at an individual nucleotide, one of those individual components, there are 5 letters total, 4 in DNA, 4 represented in RNA, then we're looking at a very kind of similar structure. Either at the structural level, you're looking at a phosphate group and a sugar. Those actually make up that backbone of the strand. And then a nitrogenous base that is either a single or a double, in this case, a double ring. In this case, it's adenine that sticks out into kind of the middle of the strand. So if you look at this, the phosphate and sugar will align with this purple backbone. So you can see when we zoom in, you see the phosphate and the sugar. That's what actually physically connects and then makes up this backbone to make the very long strand. And then kind of hanging out into the middle so that it can adhere to the adjacent strand. But remember that this is not a physical adherence. It's actually a hydrogen bond. So it's more of like a magnetic attraction than it is actually a physical adherence, which makes it easy to separate these when we need to replicate these strands, which we'll talk more about. And so you have these base pairs, either a double ringed or a single ringed base pair that then adhere to one another through those hydrogen bonds, okay? And so you have guanine and adenine, and they associate with cytosine and thymine, respectively. And you'll notice that you always have one double-ringed, base, nitrogenous base, associated with a single-ring nitrogenous base, so that they always are evenly aligned. And actually, sometimes the cell will actually detect problems with replication or problems with the DNA if there are any bumps where you end up with 2 double rings or 2 single rings. So you'll get a bulge or a kind of an indent, okay? We won't talk about that so much, but it does give this very even structure to the parallel strands, okay? So as you have these strands coming together, where you have the phosphate groups and the sugars aligned with via the backbone or in a backbone. And then you have these nitrogenous bases that are parallel to and adhere to one another through hydrogen bonding. Then you make these very long strands, and you can see here how you kind of have the backbone is this blue part. Those are your phosphates and sugars, and then your nitrogenous bases in there. And that very long strand comes together in a helical form, and then that helix can be unwound for you actually to perform replication, transcription, and translation. Okay? Now, as we said before, all cells must have DNA in the form of at least one chromosome. Okay? It could just be one chromosome. It could be multiple chromosomes. Most prokaryotes have just one chromosome, but you do have exceptions where you have organisms that have multiple chromosomes. Okay? Whereas eukaryotes tend to have multiple chromosomes generally. You could also have additional pieces of genetic information called plasmids, which we'll talk about in a minute. Okay? So first, let's look at a chromosome. Prokaryotic chromosome is going to have that long DNA strand, but it has to be stabilized. Prokaryotes, eukaryotes, doesn't matter. Those very, very long strands have to be stabilized somehow and organized somehow. I mean, if you just think about the old phones that actually used to have a cord, and they're kind of like a helical cord, we would have to do a lot just to keep that from getting tangled or whatever. And so imagine this very, very thin helix that is meters long, and sometimes multiples of them within a cell. They really definitely have to be organized. Okay. Bacteria have the most simple method for organizing their genetic information. This is a bacterial cell that's just been gently probed or poked, so its genetic information is released. And you can kind of see that the DNA spews out in these loops. And in fact, inside of the cell, that's how they're organized. You have proteins and small pieces of RNA that stabilize loops of DNA in about 50,000 to 100,000 lengths of base pairs, and then kind of aggregate and stabilize them together in loops. Whereas archaea are a little bit more complex and actually a little bit more similar to eukaryotes in the way that organize their genetic information, where the DNA, you can see this yellow piece of DNA is a double helix of DNA. And they're actually arranged around these globular proteins called histones. And histones look very similar to the way in which eukaryotes are organizing their genetic information as well. But this just essentially allows for that genetic information to be well aggregated and carefully organized and coiled, especially when it's not in use in the cell. And then you can have proteins that very specifically unwind a specific region within the genetic information so that a gene can be copied, or unwind sections of the DNA so that it can be replicated, and then wind them back around these histones so that they can be organized. Now, in eukaryotes, you have very similar to histones. Instead, these are called nucleosomes, but they're a little bit more complex than a histone, but you essentially winding your genetic information around them and then looping that genetic information until you have these X-shaped chromosomes. Okay, and so that very small X-shaped chromosome that you see on a karyotype is actually meters long, but it's very well organized. Okay, and so you can kind of see prokaryotes, even though they're small and simple, they have ways of organizing this large amount of genetic information. Bacteria in the very most simple way, just series of loops. Archaea a little bit more complex, and then eukaryotes have a really complex way of organizing all of that genetic information. Now, the genetic information in prokaryotic cells is different from ours in that it's haploid. So in a eukaryote, a human, for example, Humans have sexual reproduction, and therefore their genetic information is coming from two sources. So you have half of your set of genetic information coming from one parent and half coming from the other parent. And so we call that diploid, where you actually gain two sets of every single gene in your genome, okay? Whereas bacterial cells, prokaryotes, they reproduce asexually. Okay, and so as we go through the lecture, there'll be a couple of times when we say like conjugation pili or sex pili, for example. But remember that these are a little bit of a misnomer because actually bacterial cells, archaeal cells, prokaryotic cells in general, are using asexual methods of reproduction. And that also means that their genetic information is just one set of genes, and we call that haploid, like half of the genetic information, compared to eukaryotes that have diploid or two sets of their genome. There are a couple of exceptions to this. If you remember back to the very large bacteria called Epilopiscium that do viviparity or have live birth, they actually have thousands of copies of their same genes. So which is interesting and still being understood and studied. So there are a couple of exceptions to this, but generally speaking, prokaryotes are haploid, i.e. they have a single copy or a single set of their genes within their genome, okay? Now, we said that every cell needs to have a chromosome, at least one or many chromosomes that hold their genetic information. But prokaryotes are interesting in that they have a lot of additional DNA oftentimes that they can utilize within their cell, they can exchange between other prokaryotes. Even though they go through asexual reproduction, they can actually share with their peers. Not all bacteria, not all archaea, but there is some capacity for that. And we're going to talk more about that in both lecture and lab. And these are often in the form of what we call plasmids. Plasmids being smaller sequences of genetic information, usually a couple thousand to 100,000 base pairs long, but not really more than that. So in comparison to the genome of a bacteria, it would be about similar to 1 to 20% of a full genome of a bacterial cell. And they tend to carry non-essential genes. So what you have to have are your chromosomes, but sometimes there's a benefit, oftentimes there's a benefit to having some of these plasmids. They tend to replicate independently of the chromosomes of the cell. Now, depending on the type of plasmid, it might actually have the full capacity to replicate itself, to generate the proteins that it needs to replicate itself. Sometimes plasmids are actually managed by the original chromosome or genome as well. It just depends on the plasmid, okay? And they tend to give to that cell, whatever it is, some advantage in survival. And you have different types of plasmids. So you have fertility plasmids that allow for the cell to actually make what we call conjugation pili. We talked a little about them when we talked about the structure of prokaryotes, but essentially they're pili that can attach to fimbriae on the surface of a cell and actually make a tube where genetic information can be transferred. Some of the types of genetic information that might be transferred either on the same plasmid as that fertility plasmid or a different plasmid that then comes in are resistance genes to antibiotics, bacteriocin genes or plasmids that essentially have in them encoded toxins that can allow for that cell to compete with oftentimes genetically similar bacteria in their environment. So if they make a pili with their neighboring bacteria and it doesn't have that plasmid, it can actually kill it. Okay, and also virulence plasmids, where that bacterial cell can then become virulent to another host, a different host, like a eukaryotic cell, for example. Okay, and so depending on what the plasmid codes for, it can essentially make that microorganism kind of give it superpowers or whatever that allow it to have an advantage in its environment. Okay. Now, in lab, we're actually going to have a series of labs where we manipulate plasmids in microorganisms. And so there's functional kind of benefits to the microorganisms themselves, but there's also kind of lab techniques and benefits that we can get from utilizing plasmids and artificial plasmids within a lab setting. And so we'll talk a little bit about that in the second part of our lecture, and we'll talk a lot about it in our lab as well. Okay, so I mentioned that. So essentially, that's the structure of DNA and genetic information. There is more information in your book, especially in terms of comparison between the different domains of organisms and how their DNA looks. Here's a summary of that. So usually our prokaryotes have a single individual chromosome, although there are exceptions to that, whereas eukarya tend to have multiples. Prokaryotes do have plasmids, depending on the species of organism and the type of organism. You might often see plasmids within their cells. Eukaryotes, in specific kingdoms, plasmids also tend to be present, but not in all eukaryotes. The genetic information in prokaryotes tends to be circular chromosomes, although you have linear chromosomes as well, whereas eukaryotes, the genetic information, the DNA, tends to be linear. In prokaryotes, the DNA is located in what's called the nucleoid. So it's kind of an arrangement or an area within the cell that's specialized in holding the DNA, but it's not really well bound in terms of membranes. Whereas in eukaryotes, you have specialized nuclear envelopes that hold the genetic information within them. And then how is the genetic information organized? In bacteria, it's just organized very rudimentarily in loops, whereas in archaea and eukaryotes, you actually have globular proteins that are holding and arranging those long strands of DNA, okay? In archaea, those are called histones, and in eukarya, those are called nucleosomes, okay? So the DNA, now we kind of know what it looks like. What I want you to do is go ahead and review that information by jumping into the third link that's in the description below. It'll allow you to review kind of the structure of DNA and what it looks like. And that's going to give you some insights into the next couple of processes: replication, transcription, and translation, which is what we're going to discuss for the rest of part 1 of the lecture. Now, the very first link that I told you I'd get back to in the description is actually a worksheet that takes you through the structure of DNA, replication, transcription, and translation, utilizing a series of videos that are separate from this video, okay? So you're welcome to just stop part 1 now and follow through that worksheet. That's what you're going to turn in for your discussion post on D2L. But I'm also going to continue this lecture and guide you guys through that worksheet as well. So you're also welcome to kind of jump in and out of this lecture and follow through with the videos and the worksheet and this lecture. Either way is fine. You won't miss like a significant amount of material because I'm essentially going to summarize each part and then send y'all out into those external links that are in the worksheet, okay? They're the same ones that are listed from kind of here on out in the description as well, with a couple of optional videos that are in this description only, okay? So go ahead and jump in to the video on the structure of DNA, and you're welcome to go ahead and just work through the rest of part 1 on the worksheet and not come back into part 1 and then come back for part 2 of chapter 7, or continue with part 1 as a guided way of filling out that worksheet, okay? Okay, welcome back. So what you saw in that first video, kind of external video that we looked at, is the structure of DNA. And you're going to continue as kind of using these videos allows you to see that 3D component that's very hard for me to get with just 2D images in a slideshow. And you can really now see how that double-stranded parallel helix of genetic information lends itself really well to replication or copying. And so the next process that we're going to talk about is replication. And the replication of DNA, no matter what domain you're talking about, is what we call semi-conservative. So those two strands that are parallel to one another, in both in terms of physically parallel, but also in the information that they hold, can be easily unzipped by a series of proteins. And each one of those original strands of DNA can be used as a template for the new DNA. And that's why it's called semi-conservative, is that it conserves one of the original strands, and then it makes a new secondary strand or daughter strand on top of it, versus just copying the double helix separately, right? And so it's semi-conservative in that way. And so you can see that represented by this diagram. The original DNA is actually unzipped, and each of the purple strands is used as a template. And then that new template can then go on to the next generation. And then those original strand plus the template then are unzipped, and you can continue to replicate the DNA in that way. Okay. When we're replicating our DNA, we essentially are doing the same process with a little bit of differences on the two strands. So essentially, when you look at the structure of DNA, you'll see that we have 3' to 5' and a 5' to 3' orientation of the two parallel strands. And so replication happens always from 5 to 3'. So you'll see that that means that replication is very smooth and kind of seamless on what we call the leading strand, and it occurs in fragments on the lagging strand, what we call the lagging strand. And so you'll see that in the video, and you'll see how those occur. But essentially, you have your stabilizing proteins and helicases that kind of come into the helix and open it up and stabilize it. Okay. And that's really essential. I mean, if you think back to just using a phone with a cord, that cord can easily become too tightly wound or kind of tangled. And so whenever you're unwinding a helix like that, you have a number of stabilizing proteins. And then you have primers that come in and polymerases that actually do the copying of the strands. And so again, since these 2D images don't do justice to the process, I'm going to send you out to the next link that's in your packet and also in the description below for DNA replication. And you'll see that how that process occurs, and then you can jump back into the lecture. Okay, welcome back. So you saw how our DNA polymerase is able to take the template and then essentially copy it to make a new daughter strand on the original strand that was there. And in that way, we get two strands that are replicated in a semi-conservative way. One is in a very smooth kind of addition of nucleotides, and one is in fragments called your Okazaki fragments. Okay, and that is essentially the replication of the genetic material within a prokaryote. Now, once we have the genetic information replicated, we're going through binary fission and we have a daughter cell now. We also want to know how that genetic information is then expressed. So this takes us back to the central dogma. Here it's called the central dogma of genetics, but it's also referred to as the central dogma of biology, which says that our genetic information is expressed by first copying it in a process called transcription into mRNA, and then copying that mRNA in a process called translation into a protein. Okay, and so the next two videos that we'll watch will be the process of transcription and the process of translation. They're actually the first and second parts of the same crash course. So transcription is about the first 7 minutes of the video, and then the remainder of the video is translation. So let's just overview that for a second. Both transcription and translation occur in 3 steps that have very similar names: initiation, elongation, and termination. So transcription is copying the DNA into an mRNA copy, and translation is copying the mRNA template into protein, amino acid strands or proteins, polypeptide strands or proteins. Okay? And so let's first look at the process of transcription. Essentially, on the DNA, you have these regions called promoters and terminators at the beginning and end of a gene. So remember, the functional unit within the DNA that can actually be expressed is called a gene. And so when you have your promoter, then that essentially kind of tells your protein complex, which includes an RNA polymerase in there, to come and bind to that site and start copying. Okay, and then the terminator says, okay, this is the end. And you'll see how it's a little bit more complex than that. You have a number of other proteins that assist in that, but then you copy that gene instead of having to take the whole genome, right? You just have that one copy of that one gene in the form of mRNA, which is still a nucleotide or still nucleic acid made up of nucleotides. So it's still not functional yet, but it is a copy that can essentially be released from the copy room or from the blueprint room or from the secret vault and actually then, copied again and expressed. And that would be the process of translation. So first, go ahead and watch the first half of the crash course video that takes you through a more 3D kind of process of transcription and its details, and then come back into the lecture video. Okay, so that's the DNA transcription link that's both in the description below and the worksheet that you're working in. Okay, welcome back. So what you saw is that you essentially on the DNA have a region of the DNA that is a gene. It has a promoter and a terminator site, and you have your DNA polymerase come through and copy that region. Okay, you have a number of other supporting factors and proteins. One that is mentioned in your book and not mentioned in the video is the Rho factor, which essentially physically binds to the end of the RNA and then allows for it to kind of stop the process. So it's part of termination. And then your result is that you have from the DNA an RNA or an mRNA copy. Okay. Now our next step is to take that mRNA copy and actually read it into a functional component. That functional component is a protein. And so at this time, we're still working with nucleic acids, and nucleic acids are made up of nucleotides, as we saw before. In RNA, those are going to be A, U, and GC, okay, components. And when we translate those out through translation into proteins, you literally read off the 3 nucleotide units as one component of a protein called an amino acid. So just like in literary kind of concepts, we have letters that will read out into words. Not all words are 3 letters, but it kind of gives us an idea of how it works. So THECAT, is the cat ate rat. Okay, and so in that same way, when we look at a sequence of mRNA, we can actually look just at what are the series of nucleotides that are represented, and we can read out an amino acid very specifically, or what are called start and stop codons. Okay, and amino acids are the units that make up proteins, and proteins are the functional kind of macromolecules that can then do the work of the cells, okay? And so the process of translation translates the nucleotide units into amino acids, and then those amino acids are just attached to one another to make what we call a polypeptide, and that polypeptide will become the functional protein once it's folded. And so the second part of that crash course video does a more 3D example of how translation occurs and gives us a hot pocket analogy as well. So go ahead and watch the rest of that video and complete that part of your worksheet. Okay, welcome back. So what you saw in that video is that translation occurs again through three processes or three steps called initiation, elongation, and termination. But in this case, you're initiating via those hamburger proteins, those ribosomes, the copying of the mRNA sequence into amino acids. So the components of proteins, and then those amino acids form a long chain, and that's through elongation. And eventually that process is terminated once you come to a stop codon. And then that long chain, or polypeptide chain, or chain of amino acids, then can go off and be folded into a functional protein. Okay. So essentially, those are the processes that we're going to focus on for the first part of chapter 7. The second part of this video, part 2, is going to go through how does a bacterial cell regulate what's going on with these different processes? What about mutations? What about some unique characteristics of how bacteria actually interact with genetic information like plasmids? And how can we use some of these ideas or processes within the lab setting? Okay, so go ahead and complete your notes package. A couple of videos that are in the description and also in the notes to help you kind of complete your notes package. One is a Khan Academy video that just puts together replication, transcription, and translation in one single video so you can see how they're related to one another. So I definitely recommend watching that. And then also just an optional, fun, relaxing video, or fun, I don't know about relaxing, but called the DNA Song, which kind of gives us a summary in song form of these processes, okay? So go ahead and, this part won't end. You can finish up your notes section, maybe take a break, and then come back for part 2 of chapter 7, where we'll talk about mutation, regulation, and lab applications. See you there.